New York.
Hey, Mark Fuller, yeah.
And we've been Donald Trump free for quite a while. So, yeah, I'm recording so that the Tuesday class can watch me on 2D. Announcement, of course, Euro Tour.
Can you cut that out from the video?
No. Not at all. So Euro Tour, of course, if you're interested in going, we do have growing interest. The trip is going. So if you are able to, maybe go home and ask your parents to say, please, on top of thousands of dollars.
So do we contact you first to let you know that we want to go, or do we just bring in the paper and the money?
You bring in paper and at least your deposit, and then your spot is reserved. And what happens is with your payment, we take you up to accounting and we deposit it in accounting, and then it goes on your bank. So that basically, we have some students, apparently students in the past, like there's one student who worked at a restaurant and they were bringing their tips every week, and that's how they paid off the trip. We just had a student who brought in a money order for $35.50. He's like, here you go. And we take checks also, personal checks. And basically, if you're paying it in installments, it'll appear on banner. And if you pay it all in full, it'll just say balance owing 0. And that's it. So just fill out the application, bring in the payment to me, and we'll walk them up to accounting and take care of it. There you go. Pictures from the Black Forest region in Germany. This is in Switzerland. And then this was a vineyard in France from when I went last time, many moons ago. This Wednesday, so tomorrow, if you're interested in going to Hart Prairie College, or you're just interested about details about Hart Prairie College, they have a 1-hour presentation. Now, some students have come and said, we have class until 12:50. I asked the presenter to stick around the classroom, and he has agreed to that. He said he has no afternoon commitments, so he can stick around. So if you have been one of the students who request that, please make sure you go after your class right away to M122 to talk to Andrew from Hart Prairie College. Quiz number 6 is posted this Friday. I had a few emails about one of the answers on quiz number 5 was wrong. That has been corrected. The grades have been updated. And hard to believe, but we are in week 9. So look at that, week 9. 10, 11, 12, 13, the end at 14. So we are getting close to the end of the semester. I know time flies when you're having fun. Now, going through the materials, I realized one of the things missing on our course outline and syllabus was the heart. We do need to go over basic anatomy of the heart. We're going to start that today. If we don't get it finished, then we'll complete it next week and then keep going on skeletal muscle. It is important to know the anatomy of the heart. Why? Because when you get into exercise physiology, you're going to talk about how the heart supplies energy to the working muscles. And you need to know also for aging in the human condition, diverse populations, actually you don't take diverse populations, but other courses, because it is more than likely, even if you're, say, dealing with athletes, you're going to deal with people who have heart issues. So it's important to know what's going on with the heart. Okay, so starting right in, this is from page 286 of the current textbook, the one with Chris Paul on the cover, not Hope Solo. So it talks about basically the muscle as an organ and gives a description also, and the whole thing is covered by the epimysium. Then you have the fascicle, which is basically a portion of the muscle, and this is surrounded by the perimysium, and then you have the muscle cell itself, multi-nucleated, so this is skeletal muscle, striated muscle, and this is surrounded by the endomysium, and it's basically, it is a very long cell with striations in it. It has stripes when you look at it under a microscope, and that's one way that you distinguish it from the smooth muscle tissue. The other way is that the skeletal muscle cells are more in series as opposed to cardiac muscle cells, which kind of connect in between those strands. In terms of the cells themselves, they are very long, multiple nuclei, nuclei again being the kind of control center of the cells, where you store the DNA, which is a genetic coding. So you think computer coding, right, consists of 1s and 0s. Genetic coding consists of 4 proteins, and given how those proteins, the order in which they code, and then the proteins that they make, the way that they fold determines how the cell exhibits itself in terms of its characteristics. It has a sarcolemma, which is a muscle fiber plasma membrane that will surround it, which is different from the endomysium. And it has a sarcoplasm. So the cell itself, if you think of the cell, it's long and cylindrical, right? And then you have the endometrium, which is kind of the connective tissue around it, but then the actual cell membrane is the sarcolemma, and then all of that kind of liquid inside is the sarcoplasm, so it's cytoplasm. So it's like this big long water balloon. If you break it open, all the water spills out. That is the cytoplasm. But it's a lot more complicated than that. And we don't have time today, but maybe later on, next week or the week after, I'll show you some videos that show some of the computer-generated images which show the complexity of the inner workings of cells. You may also have glycosomes for glycogen storage. So glycogen is basically, and again, we're kind of going into physiology here, sorry. But glycogen is basically just glucose molecules stored in a chain. It can be hundreds and hundreds of glucose molecules together. So you have these glycosomes, which store the glycogen. And the glycogen is what provides energy for the cells. You also have the myoglobin. And myoglobin is like hemoglobin. So hemoglobin is in your blood. Myoglobin is within your cells. So when the hemoglobin cell passes by the muscle cell, the oxygen transfers into the muscle cell onto the myoglobin. And the myoglobin has a much stronger attraction for oxygen than hemoglobin, so it sucks in the oxygen and then carries the oxygen to the mitochondria or to the working parts of the cell. And then you have modified organelles. So you have myofibrils, which are the actin-myosin contractile proteins. You have the sarcoplasmic reticulum, and you have the T-tubules, which are involved in the contraction process of muscle cells. I'm guessing in physiology you have or are going to go into detail about the muscle contraction cycle. Yes? No? Yeah? Okay. So you should be, and that will be reviewed in exercise physiology. So you do need to be familiar with that process. The myofibrils itself. So the myofibrils are basically densely packed rod-like elements. And one muscle fiber can contain thousands and thousands of them. These are the working elements of the muscle cell. These are what cause the muscle cell to contract and get shorter. Okay? And we're concerned about this in fitness and health promotion because basically what you're doing is, in this program, you're looking at human movement, whether it's physical activity or sports-related human movement or just rehabilitation. You're looking at human movement and how human movement can benefit the organism, the human as a whole. Right? So sports performance may be how can the football player run a faster 40-yard dash or do a better team agility test. How can the powerlifter lift more weight? Rehabilitation, how can the cardiac rehab patient get up out of a chair and be mobile to get around within their house and to basically live independently. So the myofibril features, a few of them listed here, striations, which basically means that they have stripes, which you can see in this cartoon drawing, which is from 282. You have the sarcomeres and the myofilaments, and then you have this unique molecular composition of the myofilaments. And we're going to go a bit more into detail, with that. The myofibrils itself are the stripes formed from the repeating series of dark and light bands along each length of myofibril. So this cartoon drawing basically shows a muscle cell with the different nuclei stained purple, and then one individual fiber, and then you have the dark A band and the light I band. And then this is basically just a blow-up, a cartoon drawing of a blow-up, where the dark red represents the myosin, the light blue or purple represents the actin. So the actin is a thin filament, the myosin is a thick filament. And just think of it that, I mean, we're going to get into more detail of this, but imagine that we are all a bunch of rowers and we all have our paddles. And we'll all be on the same side. Probably camera can't see me, but that's okay. So everybody get a paddle ready. Come on, team, get your paddle ready. Okay, you guys get your paddle ready also, because we're working towards each other, right? And what we're going to do is we're going to paddle on the thin filament, which is the desk, and everybody reach 1, 2, 3, and go, right? And then we go, right? And then we go, right? And through that process, which you learn, oh, you guys are fired. For that process, you're like, we don't even know how to. Have any of you guys ever paddled a boat? Yeah. Yes? Come on. Oh, jeez. Okay, we'll rehearse. Okay, rehearsal starts. Friday at 3. So by basically those cross connections occurring, I can't remember the term right now, sorry. What we're doing is we're basically pulling the myosin together, and your Z discs, sorry, your H, not your H zone, sorry. So basically the M lines are being pulled together. We're going to have a cartoon picture which shows a contracted versus a relaxed skeletal muscle. The sarcomere is the smallest contractile functional unit of the muscle fiber, but not really, but it is in terms of muscle contraction. So for our purposes, if I were to say, hey, what's the smallest contractile unit of the muscle fiber? It's the sarcomere. And it contains an A band with half of an I band at each end, as shown in this cartoon drawing. And basically, they just align end-to-end like box stars of a train, and again, they just contract together. So the key thing is, through all this, the only thing a muscle can actively do is contract. You need gravity or an opposing force in order to elongate that muscle. Okay? In terms of learning all this information, I mean, we're kind of like, we're going through it quickly, but what you need to do is you need to follow this along in the textbook or on the notes, and then go home and review it and just understand what all the different terminology is, because as you learn the terminology, you'll understand how they interact with each other. Also make sure that you are able to identify the I band versus the A band, and the H zone versus the Z disc, and the M line on, say, one of these cartoon drawings. Hint, hint, wink, wink, nudge, nudge, say no more.
What's the difference between the H zone and the M line? Aren't they the same thing?
So yeah, no, they're not the exact same thing. So the H zone is basically the end of the actin to the end of the next series of actin, whereas the M line is like the anchor point for
So the M line
the
between
myosin.
and the H zone.
Yeah, so the M line is the anchor point for all the myosin. So if your myosin, this is the M line.
And the H zone.
No, because the H zone is, let's say that the three of you are the myosin, and this is the actin. The H zone is between the end of one actin set and the end of the next actin set. Whereas if I'm the anchor point of the myosin, sorry Paige, if I'm the anchor point of the myosin, I am the M line. And then let's say that chain would be anchored on this side. But he's busy eating right now because he needs his ATP for contraction. Does that make sense?
I know.
The M line is like in the H zone. The H zone is in a space.
If this is your anchor point for your myosin, that's your M line. And then if you have your actin here and your other set of actin here, the H zone is the space in between the end of the actin to the end of the actin.
Yeah.
So it's a zone as opposed to a line in the anchor point. Does that make sense?
Yeah, I just meant the M line is in the H zone.
You'll get it. Just concentrate. The myofibrils, so are basically myofilaments, which is orderly arrangement of actin-myosin filaments within the sarcomere. So now you can see, this is from page 282, you can see a bit more example where they have the cross bridges on the myosin and the actin, which is a thin filament, kind of like a rope or like the desks, right? So the cross bridges, that was the word I was looking for. Cross bridges are basically like the paddles that we use to advance along the actin. Last you. And then what each diagram here is showing if you were to cut a cross section, basically what we see here is we see just the actin. Here in the H zone, we see just the myosin. At the M line, you have the anchor points. And then basically where you have the actin and myosin, you see each myosin is surrounded by actin filaments. So it's not just the two-dimensional like paddles going out here. Again, if my stellar paddle team were to do it, right? Some of them would have paddles down here, some of them would have paddles up here to get the actin up there, some of them would have paddles up here to get the actin up here. So we're surrounded by actin. The actin filaments are the thin filaments, and the myosin filaments are the thick filaments. So you need to remember that myosin is thick, actin is thin. And by the way, keep in mind that this is, in this course, in this program in first year, this is introductory myosin, this is introductory anatomy and physiology. So this is microscopic anatomy, like very microscopic anatomy. And this is just like kind of dipping your toe in the anatomical pond, because we're talking about myosin and actin, and right now you're thinking, okay, myosin thick, actin thin. But there's also troponin and tropomyosin, which you might learn about in physiology, and we'll definitely learn about in exercise physiology next semester. And then not only that, there are different types of myosin. And even within there's cardiac myosin, glial myosin, there's within the heart different types of myosin. So again, we're just kind of like dipping our toe in the lake. And then the cross section, basically you have one thick filament surrounded by six thin filaments. So again, the black here was the myosin. You have the black here of the myosin. And then